Okay, let us learn. All of you, good morning. Yes. Okay, then let us study some just fundamentals to recall genetics and some part of the genetics, and also some things that we need to recall in genetics part, molecular biology. Just some of the fundamentals I am going to discuss. Okay, then come back to the story. There is a word called genetics. See, here I am going to touch only those components which students are often confusing. They are reading that, but they could not able to understand that line perfectly. Only such kind of things I am going to touch. I am not going to touch all the details regarding to that. If I touch all the details regarding to that, it will take lot of time. Since we have a limited hours, we will complete that one within fundamental rules. Okay, observe genetics. Genetics is a branch of science which deals with inheritance and variations. Observe, what do you mean by inheritance and variations? Remember, the process of transmission of characters, inheritance. If you talk about that, the process of transmission of characters from parents to progeny, parents to progeny, that phenomenon is called as inheritance. Inheritance means, in simple terms, remember, I could see a character in the parents. That character is also expressed in the progeny. That I can simply understand the character has been inherited. Is it okay? So, inheritance is the basis of heredity. Basis of heredity. What do you mean by that? What is first of all heredity? The characters right now what we have in our body, these are all are due to inheritance. What we have is heredity. How did you get them is called inheritance. Next one. In genetics, we will also study variations. Variations. When you talk about variations, variations are the source of evolution. Of course, if the organism is evolving, definitely that organism needs some sort of variations. How variations we develop? If you see there, variations are developed mainly due to recombination of genes. Recombination. Remember, recombinations are developed due to generally crossing over or independent assortment. Independent assortment. If you go in detail, crossing over occur in Pachytene stage. Pachytene. Independent assortment occur in metaphase 1 of anaphase 1. Altogether, these recombinations are regular variations. Regular variations, which means in every organism, recombinations are common. Whether an organism like or whether an organism doesn't like, it doesn't matter. Always in every organism, this phenomena called crossing over and independent assortment definitely occur. But there is another mode of thing is there, mutation. Mutations also causes variations. The variations that are developed by mutation are irregular. They are irregular or random variations. Random variations, which means what? We have confirmation that always there is a recombination because of crossing over and independent assortment. That's regular mode of variation. But the variations even develop due to mutations. Mutations are sudden changes. When the mutations are happening, the mutations are going to lead to development of some sort of variations. But variation that could be developed by mutation, you cannot study. Why we cannot study? Whether they will happen or not, we cannot able to understand. But variations that are developed by crossing over and independent assortment, they occur every time when there is a gametogenesis, so that we will have one clarity. The variations are developed due to recombination or crossing over are the main source for development of variations. In long story short, observe whatever the variations that you wanted to study in the name of inheritance, we will always consider the variations that are developed by recombination or crossing over or independent assortment. We will never study the mutations that are developed due to random changes. You got me? This is what we can call it as introduction of mutations. Then observe here, they are talking something called Mendel, and he has selected 7 characters. He selected 7 characters. For each character 2 traits, only 2 alternate form of trait. Be careful, trait is a character. A character may have many traits, which means, observe the statement. Imagine when I talk about a character, which means height I am talking. Height is a character. Tall is a trait. Dwarf is another trait. That means if you take one character, for one character, there may be so many traits we can able to identify. Of course, all the traits we cannot able to allow them to enter in an individual. At least maximum number of trait, 2 traits can able to enter in an individual. Is it okay? So that when you talk about character will have variations. So the variations in the character we call as traits. He had selected 7 character. For every character, 2 traits he had selected. For each trait, 1 plant. For each trait, 1 plant. Total number of plants he selected 14 plants, 14 pea plants, and he made them in the form of pure lines. What do you mean by pure lines? Genetically, they must be homozygous. And how do you know they are genetically homozygous? Upon selfing, they should not give any sort of variation at all. For example, I have taken a tall plant and I have selfed it, and it has given tall, dwarf both. This tall is not pure tall. This tall is called hybrid tall. Got the idea? This is about pure lines are always in the form of homozygous. This is, imagine, you have to remember what are the characters are reproductive characters, what are the characters are vegetative characters. For example, out of 7 characters Mendel selected, only 1 is called as vegetative character. That veg character is tall and dwarf, nothing but height of the plant. Remaining flower. Flower is a body of reproduction, especially sexual reproduction. Flower color, flower position, seed color, seed shape, and also fruit color and fruit shape. These are all are reproductive character. There is only 1 character, that is vegetative character, that is height. Then after that, go to what the Mendel had done, the crossing. You have to remember this one, okay? Because observe, I have seen another thing. Stem height is a character. These 2 are traits. Like that, flower color, violet and white. You see their pod shape. Inflated. Inflated is nothing but swollen. You know, these are members of Fabaceae members. The Fabaceae members are having marginal placentation. If the fruit is enlarged and swollen, that's called inflated. Sometimes fruits are also going to be constricted. These kind of fruits, around every seed, there are some constrictions have been developed. Because of the development of the constrictions, we are having something that is called as constricted. Constricted fruits are recessive, inflated fruits are dominant. That much remember enough. Then go to Mendelian cross. Observe this. Mendel had conducted monohybrid cross. First of all, what do you mean by monohybrid cross? Say, he wants to study single character. How? One character would be inherited. Single character. First of all, he had chosen 7 character. The beauty of Mendel studies is he don't want to study all those characters at a time. He wanted to study one character each time. For example, he had selected height of the plant. Already in the height of the plants, okay, already in the height of the plant, he had selected one is tall, another one is dwarf. He had taken a tall plant and crossed with dwarf. Then in F1 generation, all were tall, 100% were tall, and he got the surprise. I have taken 2 traits. Out of the 2, one could be able to be expressed, another one cannot express. Then why one is expressed, another one is not expressed? That is another explanation. First of all, here tall dwarf, tall express, dwarf is not expressed. Now in this tall plant, whether the dwarf phenotype is present or not, I have to know. Yes or no? For that, he had done something that is called selfing. When he got selfing, then he got the result little bit differently. That is, he got 3:1 ratio of tall and dwarf. What is this 3:1 ratio of tall and dwarf? 75% of the plants upon selfing of F1 were tall and 25% are dwarf. Then he got the tall and dwarf both are produced as progeny from only tall plant. That means this tall is not pure tall. This tall is hybrid tall. Hybrid tall. Why are you saying it is hybrid tall? In this tall plant, dwarf nature is also there, or dwarf phenotype is also there, but it could not be able to express. Then he has given the phenotype that it has expressed in F1 generation, he named it as dominant. The phenotype which could not be able to express it in the F1 generation, but it is expressed in F2 generation, he named it as recessive. Now, observe, why did you call this cross between a pure tall and a pure dwarf a monohybrid cross? Simple. If I cross between tall and dwarf, I am going to get a tall plant. Is this tall is hybrid tall or pure tall? Obviously it is hybrid tall. That hybrid is hybrid for 2 parents. This parent has inserted their genes in this organism, but one parental phenotype could be able to express, another parental phenotype did not express. That's why they are calling it as hybrid. So this hybrid is produced between the 2 different plants for one character study. That's why they named it as monohybrid cross. Is it okay? This is why we call it as monohybrid cross. Before going to the details of the further character monohybrid cross and their ratio, dihybrid cross and their ratio, etc., let us see. Imagine a situation. I have chromosome which are carrying genes. I am taking a pair of chromosome. I am taking. When I take a pair of chromosome, on this pair there are genes. Let me take capital A, capital B, small b, capital C, small c, capital D, small d. I have taken. These are the genes. Observe here the story. Imagine when we talk about genes and the alleles differences. Imagine capital A, small a are alleles. Capital A and small a are alleles. Be very careful. When I take between A and B, always you have to mention as non-alleles. They are non-alleles. What the definition that you can provide for alleles? Simple. As you say, a character is having traits. If a character is having traits, a gene is controlling character. Traits are nothing but what? Variations in character. Am I correct? Variations in the character are called as traits. Gene controlling character. Character has variation, so that the gene also must be having variations. The variations in the genes we call as alleles. Am I clear? The variations in the genes we call it as alleles. So capital A, small a are alleles. Be careful, capital A, small a, both are controlling same character. If they control same character, then only I will call them as alleles. Observe, if I write capital B, small b, for example, capital B, capital A I have written, and I am saying they are alleles. That is wrong. They are not alleles. They are genes, two different genes. Are you listening me? So, when you talk about alleles and non-alleles, observe this. In this case, capital B, small b are alleles. A and B are non-alleles. What is the definition that you can give for an allele? Alleles are same genes with slight variation. It's not compulsory for two alleles, always variation should be there. Think about that. I have capital A, capital A. Are they alleles? Of course they are alleles. Capital A, small a. Alleles? Of course they are alleles. Small a, alleles. In this, both alleles are same. Both alleles are same. One allele is working, another allele is not working. Like this, if two genes that are controlling same character, I have to call them as alleles. Got it? Then observe the story. Whenever we take two alleles, if you want to define them as alleles, they must be present on same locus. That's why the definition has given the two alleles of a gene present on homologous sites on homologous chromosome, which means they are present on same chromosome and they are present on same locus on the chromosome. What do you mean by locus? The site on the chromosome, we call it as locus in technically. So whenever we talk about same kind of genes, they present on same locus. So the same genes which are present on same locus and controlling same character, we call as alleles. So that always alleles present on homologous sites on homologous chromosomes. If they are not found in homologous chromosome, okay, if they are not found in homologous chromosome, we should not call them as alleles. Are you catching the idea? For example, capital A, cap small b, I have mentioned. This is allele to this. If I say, is it correct? Definitely wrong, because they are present on two different locus. So this much clarity you need to have regarding to alleles. Next one. When you take next concept, there is an entry of observe one statement: dominant and recessive. Observe, I am going to give definitions of dominant and recessive. Imagine you said something, okay, which is dominant and something that is recessive. Keep it in mind. Dominant, recessive, these are the statements that are often confused. For example, what do you mean by dominant? If somebody asks, I say capital A is dominant, small a is recessive. Wow, what an answer. Capital letter is dominant, small letter is recessive. That's not the definition of dominance. Dominance means the allele which can able to expressing one. That means it can control phenotype. Recessive, the allele which cannot able to be expressed. That is called recessive. The allele which is expressing is called as dominant. The allele which cannot able to express, that means the allele which has lost its ability to express, you have to call it as recessive. Think about that. I will give one thing. Tall, dwarf. Tall, dwarf. Now tell me, what is dwarf? Dwarf is nothing but the one which is not tall. That is called dwarf. Observe the statement carefully. There are no two genes, one that controls tallness, another control dwarfness. No, there are no two genes like that. There is only one gene which can control tallness. If that gene has lost its ability to express, we will call it as recessive or dwarf. Observe. Capital T is able to be expressed, small t cannot able to express. I wanted to make capital T to become small t. What is the meaning that? The one which is expressing, I want to make it is not to be expressed. The process of conversion of the one which is expressing not to be expressed, that is called as mutation. Mutation. That means, observe this. A simple example we can able to understand this. Imagine there are fair color skin individuals and there are dark color individuals. This much clarity we have. The pigment melanin responsible for colorization of skin. Careful, when we have dark skin individuals, they have melanin, that is dark color pigment. When we see fair skin individual, do you think they have white pigment? They don't have white pigment. They don't have melanin pigment. That means only pigment that is responsible for colorization of individual is melanin. If melanin present, dominant. If melanin is absent, recessive. What is recessive? Come on. Lack of dominance. Lack of expressibility is called as recessive. Having expressibility is called dominance. This much clarity we need. Are you getting the statement? Imagine a thing. I will take a flower that is violet and the flower is white. I am saying violet is dominant, white is recessive. Now tell me, how did you get the white flower? If a flower that could not be able to establish in the form of violet, that flower is called as white flower. White is nothing but no color. If color is not there, that is called white. Which means to produce white color flower, there is no white pigment would be added. There is no pigment for white color at all. Lack of any pigment, we will call it as white. But there is a pigment for violet colorization. If that pigment is there, I will call it as dominant. If the pigment is absent, I will call it as recessive. Hereafter, remember, wherever we use the word dominant, don't apply your 10th class knowledge, that is, this is capital T, this is small t. Capital T is suppressing small t so that capital T is dominant. No. There is no such kind of thing called suppression, etc., etc. What is small t? It is not able to express. Nobody is suppressing no one. Capital T can able to express, small t cannot able to express. The one which is able to express is called dominant. The one which could not be able to express is called as recessive. That must clear. Hereafter, wherever you come across dominance, the one which is able to express is called dominant. The one which could not be able to express is called as recessive. These things you have to know. Then, after that, let us take here. There was a person, see, one thing. The things that we need to consider in genetics. When Mendel was giving foundation to the genetics, at the time, Mendel did not give any kind of genotypic proposals. Genotype, right now we are learning monohybrid cross genotypic ratio 1:2:1, dihybrid cross genotypic ratio, something like that we are learning. There is no concept of genotypic ratio when Mendel was proposing loss of inheritance. Later, one scientist has come called Regional C. Punnett. Somewhere else his name is there. Okay, this Regional C. Punnett, he is the one who introduced identification of genotypic ratio. Are you getting the idea? Identification of genotypic ratios were given by Regional C. Punnett. After Punnett coming to the picture, then he provided these symbols: capital letter for dominant, small letter for recessive, and always you have to choose between dominant. If you have chosen A, recessive you have to choose always small a. These rules and regulations were given by Regional C. Punnett. Okay, then after that, same monohybrid cross which was conducted by the Mendel, we are conducting in order to know genotypic and phenotypic ratios. Let us see. We have taken pure tall, pure dwarf, F1 generation 100% tall are capital T, capital T, capital T, small t, and small t, small t. You remember these ratios. Later we will calculate some fundamental things regarding to that. This is genotype, tall, tall, dwarf. In that 1:2:1 genotypic ratio, and 3:1 it is phenotypic ratio. These ratios, careful, phenotypic ratios were given by Mendel. Mendel said 75% are dominant, 25% are recessive. Of course, it is nothing but 3:1 ratio. Later, genotypic ratios were provided by Regional C. Punnett. Whatever the genetic cross that we typically perform, that would be the combination of Mendelian pattern of phenotypic determination and also Regional C. Punnett pattern of genotypic determination also. Is it clear? This is, we need to remember. We will use some of them in order to identify. For example, often I have seen one question: how many members are there here in this one? Homozygous tall. Homozygous tall. Homozygous tall should be 1/4. Heterozygous tall should be 2/4. Is it okay? Are you getting the idea? Among the tall, how many members are? Among the tall, how many members are homozygous tall? Total members are? Total tall is 3. How many members are homozygous tall? 1/3. 3 members are showing tallness in monohybrid cross. Out of that, 1 member is having tallness, that is 1/3. Such kind of typical questions they are asking. Next one. Let us check. Then this is another kind of thing. Why they have given this kind of binomial equation here? Imagine, whatever the rules and regulations given by the Mendel, they are on the basis of rules of mathematics. Mathematic rules, if one rule is followed in basic level, the same rule must be applied in higher level also. Let us consider what is that basic level rule. When I take capital T, small t, during at the time of gametogenesis, capital T will enter into 50% of the gametes, that is 1/2, and small t will be enter into another 50%. Is it okay? Now, otherwise, tell me one thing. I want into this gamete small t. What proportions you can suggest? The probability of small t to be enter in this gamete. See, simple. This is a chair. 2 members are walking around the chair. And what is the chance small t would sit in the chair? Of course, 1/2. Is it okay? So, the probability of small t would be entered into the gamete is 1/2. The probability of capital T would be entered into the gamete is again 1/2. That means in any one particular time in a gamete, either capital T can enter or it may be enter into small t. Either one can enter. This is the two gametes that are able to enter inside a gamete. Probability of each gamete is 1 by 2. Are you getting the idea? So let us take this value never going to change. That means even you take n number of gametes, those values are not going to be changed. That's why simple. I'm going to take capital T ax plus by binomial equation. Here A is probability of capital T, that is 1 by 2, and x is capital T, plus probability of small t 1 by 2 small t. Imagine here one component I have taken, I will multiply it into 2 times. I will get the result is 1 is to 2 is to 1. I will multiply it into 3 times. I will get dihybrid ratio. I will get into 3 times. I will get tetrahybrid ratio. Like that, n number of times you can multiply, the results are going to be same. They are not going to be changed. In order to notice by you, there is no change in the result in between monohybrid cross and dihybrid cross. They have proposed this one. How do the result is not going to change? Let us solve one problem. Imagine what is dihybrid cross phenotypic ratio. If you want to know dihybrid cross phenotypic ratio, we might be by hearted. That values we have by hearted. But instead of by hearting, how to do? I know monohybrid cross phenotypic ratio, that is 3 is to 1. Since they are following binomial equation, monohybrid is one factor is 3 is to 1. I want 2 factors result. Let us multiply 3 is to 1. Then we will get 9 is to 3 is to 3 is to 1. 3 into 3 9, 3 into 1 3, 1 into 3 3, 1 into 1 1. This is dihybrid cross phenotypic ratio. Let us calculate trihybrid cross phenotypic ratio. Could you calculate trihybrid cross phenotypic ratio? 3 is to 1 power 3. Already 3 is to 1 power 2 I have calculated. Then I will make it into 3 is to 1. N number of crosses you can find out their genotypic and phenotypic results, because these results are following binomial equation. When some numbers are, be careful, this allele entry into the gamete, it does not going to change, it is constant. This allele entry into the gamete does not going to change. Is it okay? When basic thing is constant, then I can multiply n number of times, the same result is going to be repeated. That's the reason why they have given this mathematical binomial equation for us. Then go to next one. Observe. Test cross. What do you mean by test cross? Why do you want to conduct a test cross? Imagine, when you talk about dominant, for a dominant, we have two genotypes. It may be homozygous or it may be heterozygous. For instance, I have taken tallness. Tall may be homozygous tall, tall may be heterozygous tall. See, by looking at an organism, what do you check? That is phenotype or genotype. Observe this word. What do you see on an organism observation? Phenotype or genotype? Obviously, we can see only phenotype. You cannot see genotype. But here, for same phenotype, 2 types of genotypes are there. For which phenotype you have 2 types of genotypes? Dominant phenotypes. So, since by looking at an organism, I cannot determine the dominant phenotype is homozygous or heterozygous, then I have one more test in order to determine whether the dominant phenotype is homozygous dominant or heterozygous dominant. Is it okay? That test is called test cross. Remember, typically we believe test cross would be conducted. Be careful. Tall, dwarf, this is I have seen many ways, and this is tall, and then we will have tall, and dwarf. Imagine a statement. I wanted to know this tall is homozygous tall or heterozygous tall. Is it okay? I wanted to know this tall is homozygous tall or heterozygous tall. What I have to do? I have to cross it with, okay, known genotype. Imagine, look at this statement carefully. I have a dominant one and a recessive one. Whose genotype exactly you know? Dominant genotype or recessive genotype you exactly know? I know recessive phenotype and I know dominant phenotype. By looking at a one phenotype, I can say this has only one genotype. Which one should be? Obviously recessive. Yes or no? Recessive is having only one genotype, but dominant may have more than one type of genotype. So simple. I wanted to investigate a dominant phenotype is either heterozygous dominant or homozygous dominant. So I will cross unknown genotype with known genotype. Whose genotype I don't know? The dominant one. Whose genotype I know? The recessive one. Take a dominant one, cross it with recessive one. It not necessarily should be a parent. But generally we talk about it's a parental. That means F1 generation or F2 generation cross with this recessive parent. Then I will call it as test cross. Of course, you can cross anyone. That means it's not necessary that always you have to cross with the parent. But if you cross with the parent, that is also not an issue. Got the idea? That's the reason why take a dominant phenotype and cross it with recessive one. Crossing a dominant the unknown genotype of dominant phenotype to be crossed with recessive phenotype, we will call it as test cross. Test cross would give two types of result. Think about that. Here they have taken violet flower. Tell me in pea plant, violet flower color is dominant or recessive? In pea plant, violet color is dominant or recessive? Of course dominant. By looking at the violet flower, could I say this violet is homozygous violet? I don't know. So I will take this violet one and cross with white. White I know. Exactly it is homozygous. Violet I don't know. It may be homozygous or heterozygous. If this violet is heterozygous, I will get 1:1 ratio of violet and white. If this violet is homozygous violet, 100% members are going to be violet. Is it okay? So out of these two, which one is test cross? Out of these two, which one is test cross? Both are test cross. You should believe that. I have seen in many cases, people are believing this is only test cross, this is just side cross. No, not like that. It is also test cross. If I have got the violet or nothing but dominant phenotype is homozygous, I will get this result. If it is heterozygous, I will get this result. Am I clear? This is about what we need to talk about test cross. Always test cross we conduct in order to determine genotype of unknown phenotype. In order to determine unknown genotype of known phenotype, we conduct test cross. Got this idea? Okay. Now after that, sometimes they call it as, this is also a kind of back cross. What do you mean by back cross? Crossing of progeny with their parent. That's called as back cross. In the back cross, if you are crossing with recessive individual, of course already we know that it is test cross. If you are crossing with dominant individual, we call it as out cross. Of course, out cross and test cross both are belongs to back cross. Be careful. Out cross does not going to give any kind of result for us. Only test cross could be helping for determination of unknown genotype of dominant phenotype. That's about this one. Remember, most commonly we are going to think what is monohybrid test cross ratio. Test cross ratio, it should be 1:1. You said test cross is this one test cross, this is also test cross, but here you are saying it is test cross ratio 1:1. Why don't you consider here? Here there is no ratio. Keep it in mind. Here one there is no ratio over there. Only one. If they are asking ratio of the test cross, you have to give 1:1. But this is also a result of test cross. Then law of dominance, law of segregation. What is law of dominance? See, the things are like this. When Mendel proposing laws of inheritance based on monohybrid cross, generally he proposed two laws. First one is called law of dominance, second one is called law of segregation altogether. When you talk about first law, that's called law of dominance, he said characters are controlled by discrete units called factors. What do you mean by discrete? Discrete means individual, separate. Why this concept has come? Characters are controlled by discrete units called factors. The concept has come because before Mendel, whoever working in the field of genetics, they used to think like this. Whatever the characters and an organism possess, all characters controlled by one body. That body generally given by God, that was theological inheritance. That body generally given by God, so that God is controlling all characters. Mendel said this, no, it is not happening like that. If you have 100 characters, 100 factors required. Not all 100 factors controlled by one factor, not all 100 characters controlled by one factor. For every character, separate factor is required. Who said this concept? First time Mendel. Before Mendel, they used to believe that if you have 100 character, 1000 character, 10,000 character, doesn't matter, all of them controlled by one body. Mendel said no, all of them have individual bodies to be controlled. I am calling them that individual body as factor. That's why this concept has come. Then factors occurs in pair. That word also given by Mendel. Since later factor is replaced by gene, and gene is also occur in pair, that gene pair they call it as alleles. Is it okay? And dissimilar pair of alleles are called as heterozygous. Similar pairs of alleles are called as heterozygous. And later, observe here, Mendel said something like this. Whenever we have dissimilar pair of factor, one dominates the other. Here dominates means not like hammering. For example, capital T dominates small t, which means what? Capital T can express, small t cannot express. That is their meaning. Okay? Now over their story, whenever we talk about capital T dominance at the time, in a heterozygous genotype, one will be able to express, another one cannot express. This concept was also given by Mendel. If you have a dissimilar pair of factors are nothing but heterozygous body, one would dominate the other. The one which is able to dominate, he call it as dominant. The one which is dominated by the other one, he call it as recessive. These are the words given by Mendel. Then by using law of dominance, we can say, how could you get in monohybrid cross 3:1 phenotypic ratio, and why only one factor will be expressed and why not both? That would be, we can notice by using law of dominance. When you talk about law of dominance, there are some exceptions are also there for the law of dominance. The exceptions are there. Codominance. Another one is called incomplete dominance. One is codominance, one more is incomplete dominance. These two are exceptions. I am not saying the law of dominance has been disproven. Be careful if somebody says law of dominance is wrong because it has some exception. That's wrong. Law of dominance is almost correct, but here and there are some exceptions have been noticed. Is it okay? There are some exceptions are there for law of dominance. Those exceptions are there, codominance and incomplete dominance. What are they? According to Mendel concept of dominance, out of two factors, one will express, another one should not express. That's a rule. But sometimes both express, codominance. Sometimes one which is able to be expressed could not be expressed completely. That phenomena is called incomplete dominance. Okay, this is about what Mendel law of dominance. Next one, law of segregation. When you talk about segregation of characters occur in anaphase 1 of meiosis 1. We know that. Now, this is also called law of purity of gametes. Law of purity of gametes. What do you mean by law of purity of gametes? So, some minutes back you mentioned that there is a condition called heterozygous nature. Heterozygous is also called as hybrid. Am I correct? Hybrid is nothing but heterozygous. Is it okay? Now an individual I am choosing who is having capital T small t. Capital T small t, this is the individual genotype. When this individual making them to segregate during at the time of gametogenesis, one gamete, that is 50% of the gametes will have capital T, the another 50% of the gametes will have small t. Here you must remember, in one, this is gamete. Actually what I want to say, this is gamete. Into a gamete, you will get one factor completely. Into a gamete, you will get one factor completely. That factor never be mixed up. That factor, for example, into this gamete, capital T half, capital small t half, you never see. Am I clear the word? Either you get capital T or you will get small t. But you will never get hybrid gametes. When I talk about the alleles, one allele will never generate hybrid in a gamete. That's why this law is also called law of purity of gamete. What do you mean by pure? Pure plants are genotypically pure means what? Generally we believe homozygous. Am I correct? When you talk about pure, pure means lack of any kind of combination of same traits or different traits. Over there, if you take one gamete, always to a gamete, either you get one allele completely or you will get another allele, another gamete completely. But in one gamete, half dominant allele, half recessive allele, we never get. That's why this law is also called law of purity of gametes. This law says that, okay, whenever, observe this, we have two alleles present in an organism together, it could express, it could not express, but both never going to mix. Mixing of genes or mixing of characters never occur whenever two alleles present together. When two alleles present together, when the mixing never happen, that's the reason why when they are segregating, capital T will go to one gamete, small t will go to another gamete. Capital T, small t, by mixing they never go to gametes. This concept is given by Mendel because Mendel says that characters never going to blend. Blending means mixing of the characters never occur. This phenomenon is called law of purity of gametes. Let us check the next one, incomplete dominance. Incomplete dominance, it says whenever we have, okay, two, what we call, two types of phenotypes, out of them, one will be able to express, another one will not going to express. This is what we have learned in the form of, okay, in the form of complete dominance. Now we are talking about in the form of incomplete dominance. What is happened? Imagine I have taken this is dominant, which is red, this is recessive, which is white. But in F1 generation I got pink. This is somewhat closely resembled to red, but it's not red. Then upon selfing you got again 1:2, 2:1 ratio of red, pink, red, pink, white. How does it possible? It is possible because incomplete dominance. Imagine this is dominant, this is incomplete dominant, this is recessive. When they analyze, what is the reason behind the development of dominant phenotype in this incomplete dominance inheritance? Here, capital R produces pink pigment, another capital R produces pink pigment, pink is going to become red. Imagine I am adding double the quantity of pink. I am adding pink pigment in the petals, double quantity. Pink intensity will increase in the intensity of the pink, we call it as red. Here how many alleles are there? Capital R, small r, only one allele is working. That allele produces pink. This allele produces what? It's not producing white. It does not produce any color. As I have mentioned in the beginning, when there is a white, that indicate there is no pigment. White is not at all a pigment to be noticed. Over there, pink, that's the pink is established. So dominant is developed due to combination of two pinks. Since here we have heterozygous, is it okay? Heterozygous, where only one gene is working, so that the heterozygous one gene is responsible for the production of pink. So altogether, when you talk about incomplete dominance, genotypic ratio 1:2:1, phenotypic ratio is also 1:2:1. Genotypic and phenotypic ratios are same in case of incomplete dominance. This incomplete dominance first time noticed in one plant and explained in another plant. That explained plant they have given in our textbook, that's called snapdragon, commonly called as dog flower, and Antirrhinum majus. All three names belongs to same plant. Explain the concept of dominance. Observe that. What do you mean by dominance in first of all? Dominance means expressibility. How does a expressing gene will become unable to be expressed? Because of mutation. Because of mutation. Mutation making the gene which is expressing, it's not able to be expressed. The gene which is able to be expressed is called as dominant. The gene which has lost its ability to express is called as recessive. Now, when the nature has given the genes, all the genes were working in our body. During the course of evolution, some of the genes have lost their ability to express. Those genes which lost their ability to express, we call them as recessive. Is it okay? What is the reason behind the conversion of a dominant allele to the recessive allele? The reason is mutation. It's not compulsory that every mutation that is happening in a gene will convert that dominant to recessive. But if a gene has come in the form of recessive, definitely it is a mutated one. I will say the statement. All recessive genes are mutated ones. Correct. It doesn't mean that all dominant ones are unmutated. A dominant one may be mutated or unmutated, but it is still working. Which one? The dominant one is still working. The recessive one, because of mutation, it has lost its ability to express. The genes which are lost their ability to express, we will call them as recessive genes. The genes which are still maintaining their ability to express, we call it as dominant ones. Got the idea? That much you remember? Fine. Then come back to the story. Functional allele, non-functional allele, we can understand all those things. Okay? Imagine a statement. When we are talking about codominance, one thing they have given in the name of codominance, that is ABO blood grouping system. ABO blood groups. ABO blood groups are controlled by a gene that is I. I gene has three alleles. IA, IB, I0. These three alleles. Generally, one gene, if it has more than two alleles, that concept we call it as multiple allelism. If you want to study two alleles, that's easy. In one individual, two alleles will be possible. But you want to study, there are more than two alleles. That is not possible in an individual. Definitely we require population. So it has multiple alleles, and we want to get number of genotypes. For that number of genotype, n into n plus 1 by 2 formula we can use to find out number of types of genotypes possible for a population. When you apply that rule, is it okay? One moment, ma. Okay, observe this genotypic formula I am going to apply. When I apply this, then I am going to get 6 genotypes over there: A, AB, B, and O blood group. O blood group is completely recessive. AB blood group is codominance. A blood group is complete dominance. B blood group is complete dominance. Like this, when you talk about codominance, whenever two alleles express together, that phenomena is called as codominance. You should remember, for codominance also, for example, I am conducting monohybrid cross. For codominance, genotypic ratio is going to be 1:2:1. Phenotypic ratio is also 1:2:1. Is it okay? This is about it. Then after codominance, we have some concept here: pleiotropy. Imagine pleiotropy. What do you mean by pleiotropy? One gene controls or expresses for more than one phenotype. More than one phenotype. That phenomenon we call it as pleiotropy. One gene, more than one phenotype. Observe here, what is that example given? They have given one gene called B gene in case of pea plant. What the B gene is doing there? The B gene produces starch branching enzyme. Starch branching enzyme. What this enzyme does? This enzyme takes glucose and make more quantity of amylopectin. If you have an idea about starch structure, starch has two forms. One is amylose, which we call it as soluble form of the starch. Another one is called amylopectin, which is called as insoluble form of starch. So altogether, the soluble form of the starch is not going to make some kind of clumps, but insoluble starch will make more clumps. Over there, observe, starch branching enzyme is responsible for making of more quantity of amylopectin. Over there, observe, if I take capital B, capital B, capital B, small b, small b, small b, capital B, round, here also round, wrinkled. This is shape of seed. Shape of seed. Observe, and size of starch grain. Size of starch grain. And the starch grain size in homozygous condition is large, and the starch grain size in heterozygous condition is intermediate, and the starch grain size in homozygous recessive condition is very small. This is intermediate, this is small, this is large. Now over there, B gene control two types of phenotypes. One phenotype is shape of the seed. The another phenotype is called as size of the starch grain. Are you getting the idea? Shape of the seed. Another one is called size of the starch grain. Am I clear for you? Now over there, two characters controlled by same B gene. Such kind of things we call it as Pleiotropy. That's what they have mentioned here. Then come back to another story that they have mentioned. Here something they mention. Dominance is not an autonomous feature of a gene or the product it has information for. What do you mean by dominance? That explanation I have given. Dominance means what? Dominance means what? Come on. I am saying capital T is dominant. How do I know capital T is dominant? Capital T is dominant. How do I know? Is there any plant which has capital T? For example, if you go in front of a pea plant and say you have a capital T because you are tall, plant will not understand that. Keep it in mind. In that case, imagine capital T in the sense dominant allele. How do I know it is dominant? If it is produce tallness. If a allele is present in an individual and that allele is producing tallness, then only I can understand it has capital T. Is it okay? Now observe. Capital T can able to produce tall. First we will understand general biochemistry behind the production of tallness. Then we will understand what do you mean by dominance autonomous feature of a gene. Imagine capital T in pea plant produces gibberellic acid. You know gibberellic acid? It's a phytohormone and it will go and bind to the cell. It will bind to the receptor of the cell and the cell will get enlarged. When the cell get enlarged, then length, that phenomena is called as cell elongation. The length of the cell is going to be increase. When the length of the cell increases, the internodal length of the stem will also going to increase. I think you have an idea. Gibberellic acid is responsible for internodal elongation of stems. How do gibberellic acid internodal elongate internodes? Simple reason. The gibberellic acid is going to elongate the cell upon binding to the cell. Is it okay? Now observe the story. Think about that. If a plant want to be tall, okay, first T has to produce, T allele has to produce gibberellic acid. That gibberellic acid has to bind to the cell and to make the cell to be elongated. Is it okay? Now observe this story. A plant is not tall. A plant is not tall. Directly I cannot blame this capital T is not producing gibberellic acid. Why? Sometimes this capital T may produce gibberellic acid, but that gibberellic acid cannot able to bind to cell, so automatically there is no elongation of cell. Is it okay? So whenever we talk about dominance, dominance means expression of a phenotype. Here tallness directly does not depend on capital T. Of course capital T should be functional. When the capital T to be functional, then only the capital T can able to make the gibberellic acid. Okay, capital T is functional. Then can you get tallness? You may get the tallness, may not. If you are not getting the tallness, that always does not depending on capital T. Sometimes it depends on capital T, sometimes it is depend on other factors also. Is it okay? If you are watching something that is called dominance, that means expressibility, it does not always depend on a working allele. Sometimes it depends on other factors also. Therefore, dominance means expression is not an autonomous feature of a gene. Autonomous feature means one gene always is responsible for expression. If that gene has mutated, expression is stopped. Such kind of gross generalizations we cannot make, because sometimes Even though the allele is working, that allele could not be able to produce phenotype. Am I clear for you? That's the reason why dominance is not an autonomous feature of a gene. That dominance depends on as much as on the gene product. Okay, as much as on the gene product. For example, what is gene product here? Gibberellic acid. That gene product has to go and work on particular area to make sure that there is an expression of phenotype. Careful, I cannot see what is happening behind the genes. I can see only phenotype. If I see plant is tall, everything is working perfectly. If the plant is dwarf, I cannot directly go and blame capital T. You are the responsible for the lack of tallness. Maybe capital T is responsible for that. Are you getting the idea? Are you following? Maybe it is responsible for tallness. But that is the only one who is responsible for the tallness. We don't have the clarity. Is it okay? That's why dominance means what? Expression of a character is not an autonomous feature of an allele. Of course, allele will be responsible for that. But along with the allele, some other factors are also inducing its expression. So, dominance is not an autonomous feature of a gene, and it has some info. That means along with the expression of gene, some other factors also must support. Is it okay? This about dominance is not an autonomous feature of a gene. Then go for dihybrid cross. So what are the problems generally they give based on this dihybrid cross? Observe, somebody wants to make some problems based on this dihybrid cross. First, what fundamental problems they will ask, I will discuss here. That is, think about that. In this double dominant progeny, how many? Double dominant progeny. Out of the total members, how many members are double dominant progeny? Simple to calculate. Double dominant, that means I have taken the dihybrid class. I need to have both phenotypes must be dominant. One is round, another one is yellow. Is it okay? Round, it is dominant. In monohybrid cross, what is the probability of round? Dominant, 3/4. This one is yellow. Yellow is also dominant, 3/4. That equal to 9/16. This is simple one. Then let us make little complicated. Double dominant, double heterozygous, double dominant, double heterozygous. Come on. Let us calculate double dominant, double heterozygous individuals. Double dominant, double heterozygous individuals. Double dominant, first of all, right? Round, dominant or not? Yes. Yellow, okay, dominant. Double dominant, double heterozygous. It must be heterozygous. Capital R, small r. It is also must be heterozygous. Capital R, capital Y, small y. Then tell me what is its ratio? 4/16. Am I correct? Number of types they are one type. But number of double dominant, double heterozygous individuals are, number of types are 1. But number of double dominant, double heterozygous individuals are 4. These 4 are there. Okay, another question. Double dominant, single heterozygous. Let us make double dominant, single heterozygous. Come on. They are asking you to find out double dominant, single heterozygous individuals. Let us make double dominant. Come on. Round, yellow. round yellow. Double dominant, single heterozygous. One must be heterozygous. Is it okay? Another one should be homozygous. That's what they are expecting from you. So this should be 2 by 4, 2 by 16, sorry, 2 by 16. This should be 2 by 16. Total number 4 by 16. They are 4 by 16, but number of types are 2. Number of types are 2. But number of double dominant, double dominant, single heterozygous individuals are going to be, number is 4, types are 2. Is it okay? Then let us see. You said there are 9 double dominant. Out of them, one is double homozygous double dominant. One is double homozygous double dominant. Double homozygous double dominant are how many? Tell me. How many members are there? Double homozygous double dominant. The entire checkerboard. If you are following, identifying them in the checkerboard, that would be taking a lot of time. That means if you are identifying them in the checkerboard, you are doing wrong. Okay? Double dominant, double heterozygous. Sorry, double dominant, double homozygous. Capital R, capital R, capital Y, capital Y. That's it. How many members will be like that? 1 by 16. This is 1, this is 4, this is 4. 4 plus 1. That is 9. This you have got there. Is it okay? This is about the most common problems. Then there I have seen some more variety of problems. That is number of monohybrids. Monohybrids in dihybrid cross. Number of monohybrids in dihybrid cross. What do you mean by monohybrid? Single heterozygous individuals are called monohybrids. Number of monohybrids in dihybrid cross. Monohybrids, okay? Monohybrid is nothing but dihybrid cross. I mean, within the dihybrid, they are asking monohybrid cross. So, number of monohybrids in dihybrid. What is monohybrid? Single heterozygous. Come on, capital R, small r, capital Y, capital Y. Next one, capital R, small y, small y. Okay. Next, again, one more variety. Capital R, capital Y, small y. Small r, capital Y, small y. How many like this? 2. How many like this? 2. How many will be like this? How do I know how many like this? 2. This is heterozygous. 2 by 4. Homozygous. 1 by 4. So totally 2 by 4. Total number is 8. Is it okay? Number of monohybrids in dihybrid cross is 8. 8 by 16. Got it? Then let us see the another thing. Observe, after the dihybrid cross, chromosomal theory of inheritance, there's a lot of theory. You study why Mendel work was not recognized for many years, the communication problems, and Mendel's work is not understood by many because he was giving mathematical equations, etc., etc. You study them. And then go to one major concept. Here you observe, I have seen many students are confusing this. That is gene, and which is gene, which is chromosome. Okay, let me explain. Pay attention somewhat. Imagine I'm taking chromosome. This is a chromosomal pair. Chromosomal pairs, what we call homologous chromosomal pair I have taken. How many? Let me take 3. In this, there are genes, capital A, small a, capital B, small b, C, capital C, D, small d, E, F. Okay, these are the genes. Now, I am taking 3 homologous pairs of chromosomes. Okay, now when you talk about the chromosomal pair alignment, observe this story. Simple one. This is a pair. This is a pair. These are what? Pairs. Pairs. Again, this is a pair. These are pairs. Who are pairs? Genes or chromosome? When I talk about theoretically here, this is a pair. These are pairs. Okay, well done. I will give one random statement like this. On a pair of chromosome, several pairs of genes are present. Again, on a pair of chromosome, several pairs of genes are present. Are you catching what I say? On a pair of chromosomes, several pairs of genes present, which means one chromosome never carry one pair of chromosome never carry one pair of alleles. Always one pair of chromosome would carry several pairs of genes. Is it clear? One pair of chromosome never carry one pair of allele. One pair of allele means one pair. Capital A, small a, it would have several. Is it okay? That's the reason why. Let us consider. I am saying chromosome and this is pair of genes. Am I correct, ma'am? Chromosomes and genes. Which occurs in pair? Chromosomes and genes. Which would occur in pairs? Which one would occur in pairs? Of course, both. A pair of chromosome, I call it as homologous pair. A pair of allele, I will call it as, a pair of gene, I will call it as allele. Yes or no? A pair of gene is called allele. A pair of chromosome is called homologous pair. Now in this, on one pair, several pairs are present. First pair is chromosome. Several pairs are genes. So that chromosomes segregate during gametogenesis. If a chromosome segregate, the genes present on the chromosome will also going to be segregate. So that when you segregate at the time of gamete, this is also same. But only the difference is independent pairs segregate. Independent pairs. Who are the independent pairs? These pairs are independent to this. Be careful, ma. Dependent pairs called linked genes. Non-linked genes are called independent pairs. Dependent pairs. Dependent means all the genes that are present on same chromosome. What do you call them? Linked genes. Linked genes. So that when you talk about all the genes that are present on the same chromosome, you have to call them as linked genes. Linked genes of this chromosome segregate independently to the linked genes of this chromosome and also this chromosome. So one pair segregate independently of another pair. That means this pair segregate independently of this pair. At the time it becomes chromosome. Next one. Independent pair segregate independently of another. Independent pairs. These pairs are independent to these pairs. So they are called alleles or genes. That's it. Is it okay? That's about the thing. Next one. Observe. There's a linkage. The Drosophila. Male Drosophila is smaller. Female Drosophila is larger. Those things we can understand. And then linkage and recombination. First of all, what is linkage? When you talk about linkage, the physical closeness of the two genes present on same chromosome, you have to call it as linkage. Physical closeness. How far they are close? Observe. I have taken here 3. Okay. Now physical closeness I am studying. If I check A to B distance, that is greater than B to C, or A to C distance is greater than A to B, and this B to C distance is lesser than A to C to E, or if I go to A to E, distance is very more. Over here, if I observe out of this, all these genes are linked genes, but whether they are closely linked or loosely linked, that would be decided by the relative distance between them. One more time. Whether they are closely linked or they are loosely linked, that would be decided by relative distance between them. What is the meaning of that? If you talk about here, distance between A and B is more when compared to B and C. I will say B and C are tightly linked genes, when compared to A and B. A and B are loosely linked genes, but all are linked. How you say all are linked? Because they all are present on same chromosome. Are you getting the statement? Okay, that's about linkage. How to understand? You cannot study linkage between the two genes that are present on two different chromosomes. Okay, up to this, keep it in mind. The time up. In the next class, we will discuss the remaining things and also some related things regarding to molecular biology. Okay, some problems I will discuss regarding to linkage in the next episode also. Okay, that's about today. Bye.